Okay, hello everybody. Welcome back to the Atheronet Cardioscope webinars. After all, I think more than 9 months of hiatus, we are back. Now in the last year, we chose to organize webinars on demand, which means that for few next months, we will have webinars that are covering topics that were requested by people who are part of the action. And today I have a great pleasure to introduce Liv Nordestgaard. She's coming from Denmark. She spent 2 years as a postdoc in Bristol, if I'm not mistaken. And she's the best person, I think, that I know to address this topic. And fortunately, she was willing to prepare a little presentation about drug target Mendelian randomization and drug repurposing, which is a very hot topic. And without further ado, Liv, please enlighten us with this topic. And then later on, we will have time for discussion and questions.
Yeah. Okay. So thank you very much for the introduction, Miran. And I'm just gonna dive right into it. This is one of my favorite topics, so I'm very glad to be able to talk about it today. So let me see. So today is going to be about Mendelian randomization for drug repurposing and drug target Mendelian randomization, which is sort of two sides of the same coin, if you can say that. But before we sort of dive into those details, we need to first understand some basis of Mendelian randomization. So that is where I'm going to start. And actually, I'm going to start even more basic. So I'm going to start with epidemiology. So what is epidemiology? Epidemiology is a study of the distribution, patterns, and determinants of health and disease conditions in a defined population and the application of this knowledge to prevent diseases. And what we have a lot in epidemiology is associations. So this could, for example, be the association between X and Y. Or it could also be the association between HDL cholesterol and myocardial infarction. So here we have a paper from 2012 that was looking into this association between HDL cholesterol and myocardial infarction. And what the authors found here was that for a 50% reduction in HDL cholesterol levels, there was a hazard ratio for myocardial infarction of around 1.5 or 2. So lower HDL cholesterol, higher risk of myocardial infarction. But the question that we haven't really answered is whether this is a causal association. So does HDL cholesterol cause myocardial infarction or the other way around, or is something else going on? And why do we care about causation? So if we observe an association between two phenotypes, this could be X and Y. This could be due to A, X causes Y, B, Y causes X, or C, something else causes both of them. And this is also called confounding. And now the question is, how can Gregor Mendel help us with this? So Gregor Mendel, he was a monk who lived from 1822 to 1884. But before I dive into how he can actually help us, I will just quickly go through some genetic vocabulary. So an allele is a variant form of a single nucleotide polymorphism, a specific polymorphic site, or a whole gene detectable at a locus. And that's what I'm trying to illustrate over here. A locus is a section of the DNA sequence and can be a single nucleotide polymorphism, a gene, or a larger region of DNA sequence. That's sort of what I'm illustrating here. And a genetic variant is a position in the DNA sequence at which there are at least two forms in the population. And that's what I'm illustrating here. So here there's one variant is CG and the other one is TA. Meiosis is a special type of cell division of germ cells in sexually reproducing organisms that produce the sperm or egg cells. It involves 2 rounds of division that ultimately result in 4 cells, each with only 1 copy of each chromosome, so they are haploid, and that's illustrated here. And dominance is the phenomenon of one variant or allele of a gene on a chromosome masking or overriding the effect of a different variant of the same gene on the other copy of the chromosome. And that's sort of illustrated here. And then we have genetic linkage, which is the tendency of DNA sequences that are close together on a chromosome to be inherited together during the meiosis phase of sexual reproduction. And I couldn't really find an illustration of that. Okay, so how can Mendel help? So Mendel, he was a monk and he studied pea flowers. And what he found out was that if he had a purple male pea flower and a purple female pea flower, when they reproduced, one likely scenario was that they had 3 purple baby flowers and 1 white baby flower. And why was this? So this was because of the dominant and the non-dominant alleles that they were inheriting. So 2 flowers would have a dominant purple allele and a non-dominant white allele, and they would become purple. 1 flower would have 2 dominant purple alleles, would become purple, and the last one would have 2 non-dominant white alleles and would become white. And while Mendel was studying these flowers, he also wrote down some laws. And one of these laws is Mendel's second law of independent assortment. And this states that during meiosis, assortment of alleles at a locus occurs independently of the assortment of alleles at another locus, provided there's no linkage between the loci. And then he also wrote down Mendel's first law of segregation. And this one states that during meiosis, only one set from a pair of homologous chromosomes is transmitted to each gamete. And how can we use this in Mendelian randomization? So Mendelian randomization, you can compare to a randomized clinical trial. So when you have a population, you have a random segregation of alleles at conception. So you will have one group that doesn't have the allele or the variant that you're interested in, and another group that has this variant. Because you have an equal distribution of confounders, you will be able to compare the number of disease outcomes in these 2 groups. When you have a randomized controlled trial, again, you have a random allocation into 2 groups, a control group and a treatment group. And because you have an equal distribution of confounders, you can compare the disease outcomes in these 2 groups. And what we use in Mendelian randomization are genetic variants, and we call them instrumental variables. And this is because instrumental variable analysis comes from economy, where they didn't use genetic variants, but they used something else to tell them something about causation. And what we can say is that if we have variants or alleles that are associated with the exposure of interest, the variation can be used to determine if the exposure causes the outcome. And this is illustrated here is the association we saw before, so association between X and Y and a confounder C. And here we don't know whether X causes Y or Y causes X, or if it's a confounder or what's going on. But then if we have a genetic instrument that associates with X but is not affected by confounders, then we can say something about whether X actually causes Y. And when you use instrumental variables, there are some assumptions that needs to be met. And these assumptions are 1, the relevance assumption, and this states that your genetic instrument has to be associated with your exposure of interest. The second one is that there should be no confounding. So you can't have any confounders that affect both your genetic instrument and also affect your outcome of interest. And then the third assumption is that you have no horizontal priority P. So your genetic instrument can only affect the outcome via the exposure and not through another pathway like what is illustrated here. And then there is also the gene environment equivalence assumption, which assumes that if you have a genetic effect that, for example, lowers your LDL cholesterol with 1 millimolar per liter, this effect and the effect that this has on your risk of disease will be equivalent to the effect if you lowered LDL with 1 millimolar per liter using a drug, for example. All right, so back to the HDL story. So here we're looking at hazard ratio and odds ratio for myocardial infarction per 50% reduction in HDL cholesterol levels. And here we have the observational estimate, and then we have a genetic estimate. And what we can see is for the observational estimate, it looks like with lower HDL cholesterol, you have a higher risk of myocardial infarction. But when you use genetic instruments that actually affect HDL cholesterol levels, there is no association. Okay, so Mendelian randomizations also has some limitations or possible causes of bias. Among these are population stratification, which is the presence of subgroups within a larger population that have different genetic structures and allele frequencies. Assortative mating, which is a mating pattern in a form of sexual selection in which individuals with similar phenotypes or genotypes mate with one another more frequently than would be expected under a random mating pattern. Then we have survival bias, that is if you have genetic variants that affect who can survive until they get the outcome you're interested in. Pleiotropy, which is a phenomenon where a single genetic variant influences multiple seemingly unrelated phenotypic traits. Sorry. And then you have weak instrument bias, that is the bias that occurs when your instrumental variable used in your MR analysis is only weakly associated with the exposure. And there are more as well. And now actually it's time for a quiz if you're up for it. Let me just see. So can everyone see my screen?
Yes, it's we can see the changes in
okay. You ready for a quiz?
Always.
Okay, all right, here we go. So if you go into kahoot.it, and then there should be a pin code coming up here. So if you put the pin code and you type it in, and you not that many participants. Oh, Miranda's there. Wonderful. Maya, Matt, nice.
I managed.
Well done. Could we have some more on maybe? All right, Everton, welcome. Roxanne, Sandra. So we're 13 right, so we're still missing some people. I think we're missing Demetrius as well.
I'm trying to enter, Liz. I don't know how.
All right.
I went into Kahoot website, but how can I join the?
It's .it is important, so Kahoot.it. All right, Amara. Did you figure it out? Bojana? Did you figure it out, Demetrius?
No. Do I have to log in to Kahoot?
Yeah. You just have to use this pin code. So Kahoot.it, and then you put in this pin code. You can also scan the code, the QR code.
Okay, let me see. Let me do this. Okay.
All right.
I did. Yes.
Wonderful. Let's go. Let's go. Okay. First question. How can Mendelian randomization help with the question of causality? Using instrumental variables, finding individuals to genotype, using genetic variants, or examining multiple associations at one time. And there can be several correct answers. And you get points for being fast, and you also get points for answering correct. Okay. You have a lot of answers there. All right, 15 answers. Yeah, well done. So quite a lot. Got correct answers. Well done. Very well done. And let's see who's in the lead. Maya. Congratulations. Okay. Next question. An instrumental variable has to be associated with the outcome independent of the exposure, associated with the outcome, associated with the exposure of interest, or confounded. This one's a bit harder. All right. Well done. Still quite a lot that got the right answer, so well done. Okay, who's in the lead now? Roxanne. Well, and Dimitra is number 2. Well done. Okay, so question number 3. HDL cholesterol levels are A, associated with risk of developing myocardial infarction, associated with alcohol consumption, a causal risk factor for myocardial infarction, or a causal risk factor for alcohol consumption. All right. Well done. A lot of correct answers here. Very well done. And we got Miran climbing up the steps. All right. Question number 4. Mendelian randomization can be biased by population stratification, assortative mating, survival bias, or weak instrument bias. It looks like we got all answers in. Yeah, and all answers are correct, so well done. Okay, still got Roxanne in the lead. So let's see, 5 out of 5. 5 out of 6. Population stratification is subgroups in the population with specific phenotypes, subgroups in the population with high BMI, subgroups in the population with different genetic structures, or subgroups in the population who live longer. So this is a hard one. All right. Well done. There's a lot of correct answers here. Very well done. Okay, so actually the last question we're going to save for later. So now we're just going to move on with the presentation. All right. Okay. So now we're coming to the thing that I think you're actually all interested in hearing about. So this is drug target Mendelian randomization. And drug target Mendelian randomization uses genetic variation in genes encoding drug targets. This could be, for example, HMGCR, which encodes statins. It can provide evidence of causation of a drug target on an outcome, and it can enhance the success rate of drugs in phase 3 outcome trials. It can help predict side effects of drugs, and it can be used to repurpose drugs to other diseases. And what is important when you do drug target Mendelian randomization is first to think a bit about the background. So biological plausibility. Is it plausible that this drug target will actually have an effect on the disease you're looking at, or are you just trying random things? Because then it's a bit too much work if you don't have a good idea about that there's actually going to be something to find here. Then it's important to think about the selection of instruments. It's important to think about the selection of exposure. You also have to think about the selection of method. You have to think about pleiotropy, because this is quite an important bias that can happen in drug target MR. And then finally, I am going to show you an example of a drug target Mendelian randomization. Okay. So when you select your instruments for drug target Mendelian randomization, you can either choose cis variants, which are genetic variants that are within or close to the protein encoding gene that are more likely to exert protein-specific effects. You can also choose trans variants, and trans variants are genetic variants that are not at or near the protein encoding gene and are more likely to have pleiotropic effects. And this is illustrated here. So here you have the cis variants that are in the gene encoding the protein, and they are likely to only affect the protein that you're interested in. And then when you look at the effect of these variants on the disease, you will get the effect of this protein on disease. With trans variants, you've probably selected them so that they also affect the protein that you're interested in, but they're more likely to have other effects, and some of these might also affect the disease through other pathways than the ones that you're actually interested in. And this is where you get a horizontal pleiotropy that will invalidate your findings. When you select exposure, you can select different exposures, so you can choose protein levels, for example. So if this was HMGCR, it would be protein levels of HMGCR. You also call that pQTL. Then you can select variants that affect the mRNA levels of the drug target, called eQTL, or we can choose variants that affect a biomarker downstream of the drug target. So if it was HMGCR, the downstream biomarker could be LDL cholesterol, for example. Then you need to think about the selection of method. And here you mainly need to think about if you want to do a one sample Mendelian randomization or two sample Mendelian randomization. And the one advantage with one sample Mendelian randomization is that you will have individual level data available. So you will know the BMI, the blood pressure, and the genetic variant of each individual. Another advantage is that you can test the association between your instrumental variable and your exposure, both in the population where you have your exposure and in the population where you have the outcome, because these are the same, because it's a one sample. If you have weak instruments, they will bias the results towards the observational results. And an advantage is that you can test for confounders of the risk factor outcome association. And another advantage is that you can do subgroup analysis. Then we have two sample Mendelian randomization. Here, A disadvantage is that you can't test the association between your instrumental variable and your risk factor in the outcome cohort. If you have weak instruments, they will bias the result towards null. An advantage is that you're more likely to have more power because you're using two different cohorts, so you will have just more individuals and more cases, most likely. A disadvantage is that you can't do subgroups analysis, and another disadvantage is that you can have some bias if there's been adjustments made in the GWAS that you're using. All right, and one thing that's important to keep in mind is pleiotropy. And here we're looking at target-mediated pleiotropy, which in drug target MR is sort of like the good form of pleiotropy. So this is features of a drug target leading to phenotypic effects that differ from those arising from the downstream complex biomarker. So this is our drug target. This could be HMG for CR, for example, and our biomarker could be LDL cholesterol. So we could see that HMG-CR would affect LDL cholesterol, and this would affect the risk of ischemic heart disease. But there could also be another pathway where the drug target affects the disease. But if the variants you choose have the same alternative pathways as the actual drug target, that's not a problem because you are interested in mimicking the drug target, and so if you get effects that you also get when you give the actual drug targets, that's fine, and that's actually what you're interested in. The problem comes when you have horizontal pleiotropy, and this is where your drug target MR can potentially be biased. So here again we have a drug target, and this drug target affects a downstream biomarker, and this affects the risk of disease. But then one of these variants, it does affect the complex biomarker, but it also has another effect that the drug target doesn't have. And this is when you will violate the MR assumptions, and you are likely to have a biased estimate. Okay, and now for our example. So this is a paper where they're trying to characterize the effect of lipid-lowering drugs on the metabolomics in UK Biobank using drug target Mendelian randomization. And the drug targets they're looking at is HMG-CR, PCSK9, NPC101, CTP, ApoC3, NGPO3, NGPO4, and LPL. HMG-CR is a drug target of statins. PCSK9 is a drug target of PCSK9 inhibitors. NPC101 is a drug target of ezetimibe. CTP is a drug target of obicetrapib. ApoC3 is a drug target of olisiran or something like that, I think. And I think the rest of them don't have any official drugs yet, as far as I know. They might have. Anyway, so the aim of this study was to investigate the effect of drug targets in risk of coronary artery disease, estimate the effect of the drug targets in risk of type 2 diabetes, and estimate the effect of the drug targets and their effect on 249 metabolic traits. So this is just to, again, show you the principle of drug target Mendelian randomization. So here you have HMG-CR, the drug target of statins. And when you want to choose variants to use in your drug target Mendelian randomization, you can choose variants that are within the gene here or very close to the gene, as is illustrated here. And these variants, they will affect the HMG coenzyme reductase, so the protein, and this will affect your risk of coronary artery disease. Okay, and then when they had to select exposures, they selected downstream biomarkers. So for HMG-CR, PCSK9, and NPC101, they selected LDL cholesterol. For CTP, they selected HDL cholesterol. And for triglyceride, and for ApoC3, NGPO3, NGPO4, and LPL, they selected triglycerides. And their reasoning for doing it like this was that the downstream biomarker was supposed to be the biomarker that these drug targets were designed to mainly affect. Okay, so they then did a GWAS of HDL cholesterol, LDL cholesterol, and triglycerides in the UK Biobank. They adjusted for age, sex, fasting time, and genotyping chip. The criteria for including variants, genetic variants, was that they had to be associated with each of these biomarkers with a p-value of p smaller than 1 × 10⁻⁶. They had to be located within 100 kilobases region on either side of the encoding genes, and they had set a limit for linkage disequilibrium of r squared less than 0.1, and this says something about how likely it is that two variants are always inherited together. And if you have two variants that are always inherited together, that in your instrumental variables, this could bias your results. Okay, then they also did genome-wide association studies of the metabolic traits. So they used the metabolomics, the NMR metabolomics in the UK Biobank, and they applied a GWAS pipeline to 249 metabolic traits. And they adjusted for age, sex, fasting time, and genotyping chip. And this is just an illustration of a GWAS. So here you have the chromosome number on the x-axis, and you have the strength of the association on the y-axis, and if this could, for example, be LDL cholesterol, and then you could see that here on chromosome 6 there was a variant that associated with LDL cholesterol, and here on chromosome 13 there was another variant that associated with LDL cholesterol. All right. So the genome-wide association studies that they used that they didn't make themselves was the cardiogram plus C4D consortium, including 60,800 cases of coronary artery disease and 123,500 controls, and the Diamante consortium, including 74,000 cases of diabetes and 824,000 controls. And for the statistical analysis, they did 2 sample MR to investigate the genetically predicted effects of each target on risk of coronary artery disease and type 2 diabetes. And then they also did 2 sample MR to estimate the genetically predicted effect of each drug target on the 249 metabolic traits. And they used the R package that's called Mendelian randomization. And now for the results. So they found 137 instruments that could be included with the criteria they had set for inclusion. And you can see here are some of them listed. So here's NGPL3 and NGPL4. Here's the RSIDs, the chromosome number, the position on the chromosome, the effect allele, the other allele, the beta coefficient, the standard error, and the p-value. And they then created scores, including these variants for each drug target, and look at the risk of coronary artery disease and type 2 diabetes. And the results are odds ratio per 1 standard deviation lower drug score. So for coronary artery disease, they see quite like what you would expect to see. So lower LDL via HMTCR reduces the risk, and so does NPC101 and PCSK9. There's also a slight reduction for CGP with the increase in HDL cholesterol MSB, and there's a lower risk of coronary artery disease for NGPL4, so lowering of triglyceride via NGPL4 and via ApoC3 and via LDL. The only one that doesn't have any effect is NGPL3. Then they also looked at risk of type 2 diabetes, and here it looks like there is actually an increased risk of type 2 diabetes when you lower LDL via HMGCR, probably also an increased risk with NPC101. And then they see a lower risk when you lower triglycerides with NGPL4 and LPL. And they wanted to investigate this metabolic signature of the different drug targets. So here they're looking at HMGCR, and they have the lipoprotein particle concentration, cholesterol concentration, and triglyceride concentration. And what they can see is that when you lower LDL cholesterol with HMGCR, you will have fewer extremely large VLDLs and, well, all the different kinds of VLDLs, IDL, LDL, and the very large HDLs, but not the rest of them. You will also see a lower cholesterol concentration in different lipoprotein particles, except for the large, medium, and small HDLs, and the same goes for the triglyceride concentration. For NPC101, the results look quite similar. So lower lipoprotein particle concentration for VLDL, IDL, and LDL, but no effect on HDL. Lower cholesterol concentration in the different particles in VLDL, IDL, and LDL, but not in HDL. And then for triglyceride concentration, it looks like there's actually a lower concentration in almost all of the different lipoprotein particles. All right, so they conclude that they can characterize the repertoire of genetically predicted lipid modifying therapies on the blood metabolome. They can demonstrate heterogeneity between the different classes of drugs, and they can predict effects of drugs for clinical development as a marker of treatment response. And now it's time for the last question in the quiz. If you haven't signed out of it, so let's see. Roxanne is in the lead. Demetrius is second. Everton third. Very exciting. Are you guys ready?
Should
be ready. All right. Last question. Drug target MR can enhance success rate of drugs in phase 3 outcome trials, predict effective DI and action, be used instead of randomized trials, help predict potential side effects. And there can be more correct answers. All right, we got a lot of correct answers here. Let's see who's the winner. Very exciting. Number 3, Maya. Number 2, Everton. And number 1, Roxanne. Congratulations. Well done. And Dimitris and Sandra are 4th and 5th. Well done, everyone. So the prize is the honor, which is probably quite a shitty prize, but I don't have any other prize. Yeah, and that was actually it for me. And if you want to do a project where you're working with Mendelian randomization, you can shoot me an email, and I'm happy to take any questions. And now I'll stop this.
Thank you so much, Liv. You really kind of enlighten us about the Mendelian randomization, but also, you know, make it a joyful journey. Friday afternoon, I think it was quite nice. I just might add that honor as a reward is most most of the time in academia something that we used to. So in that sense, we are doing good. Thank you so much. I think this is very, very important topic. Because it addresses a use of an instrument that is currently very available, because we have a UK biobank okay, we need to pay a little bit or a little bit more to have access on it. But
Yeah.
based on the data that is already out there, we can already stratify our patients and see the causality of our targets with specific outcomes. What I'm surprised about the results that you show at the end is that inhibition of HMJ coenzyme A reductase is quite associated with diabetes, if I understood correctly, which and not only HMJ coenzyme A reductase, but also with PCSK9. So the whole theory about using statins in the prevention of, I mean, going with using statins to treat patients in early age to kind of avoid the development of coronary arteries disease might be overthink after this, at least in a sense that we need to know more about the interactions and why in some cases this is the reason. But again, if this is done on a large population, it seems that it's an effect that is not dependent on something else, but probably just on this. So that was for me very, very interesting observation from that study.
But I think it is known, I think it is known that one of the side effects of statins is to increase glucose, right?
Yeah, this was known, but I never saw this, shown in such a convincing way. I would say that was always either the speculations or, there are lipophilic statins, there are hydrophilic statins, this influence more, this influence less, but I never see this very convincing argument from the genetic perspective that actually changes in HMJ coenzyme A reductase can induce directly the very great risk of diabetes. For me, what I find kind of challenging to understand is how we explain, when we can come back to your example of the HDL, how we can explain that, we see this causality, which is not, we see the association of HDL being associated with heart disease, but then there is no causality there because, traditionally when we teach these things, there are Tangier disease and all these things that are associated with very low HDL, and then there is great risk of coronary artery disease. So what is the missing link? What is the factor C? What is the co-found? We don't see.
Yeah, I'm not sure about that, but the thing is that when you have low HDL cholesterol, right, you quite often have high levels of a lot of the other lipoprotein particles and cholesterol in VLDL, LDL, and so on. So I think it's definitely some confounding in that. And what's going on with Tangier disease? That's also an interesting case because that's an even more extreme case where you have extreme levels not only of HDL but of a lot of different lipids, right? So yeah, there's definitely something we don't understand, and also there is always bias when you do Mendelian randomization. So it's not like it's always a holy grail, but for example, with CNTP inhibitors, what you saw is that when you had the CNTP inhibitors that only raised HDL cholesterol but didn't affect the other lipoproteins and the cholesterol concentration, there wasn't a reduction in coronary heart disease, right? But then when you have CNTP inhibitors that also lower LDL cholesterol and cholesterol in VLDL and so on, they actually had an effect on preventing coronary heart disease. So yeah, you always have to be a bit skeptical and look for bias, look for confounding, and like try and see it from different perspectives, I think.
Let me check if we have any questions from the audience in the chat. Dimitris, if you want, you can ask something.
I don't think there are any questions in the chat. No, I don't have any questions. That was a great seminar, Liv. Thank you very much.
Thank you.
Many people don't know what Mendelian randomization is, and they hear about it a lot in papers and in conferences. So I think your presentation was very, very clear. So
thank you.
It helps a lot.
Yeah, it was nice to see. I mean, still quite a lot of people, or like 15 or something. That's quite good. Yeah, I was happy with that.
We had 22, 24 at some point, which is good.
Oh, nice. Okay, that's very nice. Yeah.
I have just one more question for the end, and then we can all go and have a Friday night.
Yeah.
There's a lot of combination between the Mendelian randomization, GWAS and TWAS, in combination to find how specific SNPs, I guess, influence mRNA levels. And at some point you actually touched upon this. So if you can comment on this approach and what it can brings to the table apart from what you already have explained.
Yeah. I mean, this is actually something that I don't know that much about, and I think I would probably be a bit cautious in using mRNA as the exposure when you're looking at a drug target, because that's not usually what we're trying to target. So, but in some cases you can obviously use it, but I think the most important thing is that you always think about what is the biological plausibility behind this, like how would this work, how would mRNA actually affect the risk of disease, not just kind of just test a lot of different things, but actually think about the mechanisms. And I'm not really an expert in mRNA, so yeah.
Okay, good. Thank you so much for sharing this with us. It was quite a pleasure and quite a joy to the quiz and everything. Congratulations to the winners of the honoring prize.
Yeah, well done. Well done. Yeah, thank you so much for listening.
Thank you.
I wish you a nice weekend, everybody. And I guess next webinar is going to be sometime in the end of February. You will receive the information about it. So, stay tuned.
Okay.
Thank you. Have a nice weekend.
Bye-bye.
Bye-bye. Bye.